So our module 2, the first one is the RNA-directed RNA synthesis. And what you will see that the rigor of the material will go up a little bit. And it is a little bit of adjustment, and that's the reason that we are just not going to rush through this one and not going to finish the whole chapter today itself. We will just keep a little bit for Thursday as well. And so being said that, you will have only one quiz due on lecture 6, excuse me, lecture 6 topic due on this Sunday, just only one chapter. But do not wait until Thursday. The material that we do or we just start today, just make sure that you start going through the material, because you need a little bit of detailed understanding of the material. And it is like a little bit of mathematics in the sense that there are very, very specific logics about what we are going to talk about in the molecular biology. And this will be really like a hardcore molecular biology. And for that reason, I just uploaded a supplementary material just to remind you some of the basic structures, some of the transcription replication that we have talked about in the supplementary material. You can look at that. There was another paper about one of the techniques called polyacrylamide gel electrophoresis, urea polyacrylamide gel electrophoresis. We analyzed the RNA fragments using those in a high resolution. And even the RNA fragments which differ in one base, we can just analyze those. So just an overview of that, not going to ask any specific question on those, but there could be some question that could be formulated based on some of those techniques where you do not need to understand the technique, but understand the experiment, and you will be able to answer accordingly. We will go over that, and you will see some of the examples of those in your quizzes, so you should be okay. So let's start with the next step. So as we said that in the first module, we finished pretty much with the attachment and the entry, and we have seen the virus has delivered, or any kind of virus delivered its nucleic acid either into the cytoplasm, depending upon what the virus is, or into the nucleus. And from that part, we will actually start the next step. And first, we were going to talk about the general principles that pretty much all viral families follow that. And then we will take the specific prototype examples from the Baltimore's scheme, and we are going to talk about some of those specific ones in detail. Although you have a general principle for each of the Baltimore class, there is a substantial difference in between the families, and it is almost impossible to talk about all the families. We will just talk about some of the few important ones as well. So the first thing is that a little bit of history, really, you do not have to memorize those, that just to put things in perspective, that 1935, Wendell Stanley crystallizes the tobacco mosaic virus, and known that it contained about 5% of RNA. So you can easily think about that almost 90 years, so many years ago, close to 90 years, this was being done. But really in 1959, RNA was identified in many animal viruses. And in the 1960s, as you can see, the studies on the viral RNA replication began. And mainly with a hallmark discovery by David Baltimore, and we are going to talk about. And this is for the fun that I was born in 1971, so I'm pretty old. And in 1995, I started working with the double-stranded RNA virus of the Rio Verde family. It's a cyphovirus, it's an insect virus. And in 2001, started working with a negative strand RNA virus of the Rhabdoviridae family, which is the vesicular stomatitis virus, VSV. So this is, I thought that just for fun, I just put it on there. There's nothing of that of important historical perspective. All right, so next what we are going to see is again, revisiting the Baltimore scheme, and we are just looking at this red box right here. And we're going to talk about this, the class 3, which has a double-stranded RNA, example, reovirus. Then we're going to talk about class 4 with a plus-stranded RNA, poliovirus. We do not cover usually coronavirus. Instead of that, usually we used to do another virus called alpha virus, but I replaced that alpha virus this time with the coronavirus. It is extremely complicated, the coronavirus replication, and that's the reason that I just put a little bit of small part of it, of that, just to put things in perspective. And then we are going to talk about the negative sense RNA viruses, influenza and the vesicular stomatitis virus, and why we have two, because there are two different things. The influenza virus will be a segmented negative strand RNA virus, where the vesicular stomatitis virus will be a unimolecular negative strand RNA virus, just have one long gene, 11 kb gene, whereas this will have 8 segments of negative strand RNAs. But many of the principles are the same. So first, we are looking at some of the original papers, and this was the first preliminary data from David Baltimore, who was a graduate student of Richard Franklin at the Rockefeller Institute, and this was published in 1962. You can see that the preliminary data about that RNA-directed RNA polymerase, RNA-directed RNA synthesis can occur, and a viral RNA polymerase is probably responsible for that. So how they discovered this, and this is a very interesting experiment that how they did. So initially they were doing their experiment with a mouse, mango virus. It was isolated in mango in Africa. And David Baltimore initially started working with that, but he was lost interest in that virus because it doesn't infect. It's not an important virus in the sense that it doesn't infect human and cause anything. It causes paralysis in some of the other mammals like mouse. So he moved from the mango virus to poliovirus, and most of the studies were done with the poliovirus. So what they did, that they took the cells, and again these are in the culture, and they added all the nucleotides, and out of those nucleotides, there is one of them in the original experiment they use a carbon-14 labeled GTP. So one of them was radioactive. And what they do, that they added, before adding these nucleotides and the hot or the radio-labeled nucleotide, they added actinomycin D into the cell culture. So what actinomycin D, that this is an inhibitor of the cellular RNA polymerase, means that DNA-dependent RNA polymerase, which actually does transcription in a mammalian cell or any eukaryotic cell, RNA polymerase. Actinomycin D will not allow that RNA polymerase to produce any RNA. So if you add the hot or the radio-labeled GTP and the other nucleotides, after you have inhibited the RNA polymerase, if anything actually happens, then there must be something different. So what they did, this right here is the same graph from the original paper, just modified a little bit. You can see that this is the hour of post-infection with the virus, with the poliovirus. And right here is the poliovirus amount. So you have the initial eclipse period, then you have the burst. That's what you are seeing, the poliovirus growth in the red. And you are seeing that RNA polymerase activity right here, which is plotted right this. So they actually argued that this polymerase, which is synthesizing the RNA, so that's what they measured right here, must be done by a RNA polymerase, which is not inhibited by the actinomycin D and must be of a viral origin. It is not a native thing in the cell. And then a number of other papers started getting published. People started working on that, and then the RNA polymerases were purified from the cells that is infected by these viruses, and also in some of the viruses where the RNA polymerase is packaged into the virus particle. And that was pretty much the prelude of the whole thing. So a couple of important things that a polymerase discovered in negative strand virus particles. So this is a rhabdovirus, vesicular stomatitis virus, or rabies virus are the thing. And you can see that these tiny molecules right there represent the RNA-dependent RNA polymerase in the particle. And it was also discovered in the double-stranded RNA virus particle, like reovirus, the RNA-dependent RNA polymerase. And we will just talk we'll just shortly say is RDRP, RNA-dependent RNA polymerase, where the RNA template is used to make an RNA. That's what it is. In eukaryotic cell, it is always DNA-dependent RNA synthesis, which we call as a transcription. So that's the difference. And we will abbreviate this as RDRP, RNA-dependent RNA polymerase. And the plus RNA viruses do not have the RDRP in the particle. And we will see that because the plus RNA is itself an mRNA, the virus codes for the RNA polymerase. So the plus RNA goes into the cell, translated, is immediately translated, made into the proteins, and one of the protein will be the RNA-dependent RNA polymerase now present in the cell. The same thing in the poliovirus, which is a plus-strand RNA virus, and that will now do the mRNA synthesis and the genome replication of the genome RNA. Whereas for the minus sense RNA virus or the double-stranded RNA virus, these are not mRNAs, and that's why they need to have the particle, they need to have the RNA polymerase present in the particle, need to bring it to the cell to initiate mRNA synthesis. There is no, you cannot translate these RNAs into proteins. So all the RNA polymerases do have this GDD. This is or close motif, and this is a glycine aspartic acid, and we are going to talk about a little bit more detail about that. And there are many functional recombinant RNA-dependent RNA polymerase studies. There are hundreds and hundreds of structures already solved by X-ray or cryo-EM, but the first structure was solved in 1985, which was the poliovirus. And poliovirus 3D Pol is the name of the RNA-dependent RNA polymerase, which was the first one to be elucidated by the X-ray crystallography. So here we are looking at that all viruses do not have the RDRP in the particle. We talked about that in the minus sense RNAs, you do have the RDRP in the particle, as you can see right here. And this is the influenza virus. Each of these segment do have an attached RNA polymerase at the end of it. You can see that. And right here, all of the minus sense genomes of the genomes are coated with a protein. There is no exception. This is a very important thing. It's already in your notes that what you're going to see, that all negative sense RNA genomes are coated with a protein, no exception, segmented or unimolecular. And that's the important part. On the other hand, the plus-strand RNA genome, there is no RNA-dependent RNA polymerase in the particle, and they have a naked RNA. They are not coated with any kind of protein, because the reason that their RNAs are mRNA. So when they infect the cell, their genome RNA comes out, and the cell actually see them as an mRNA and do not attack them. Whereas we're going to talk about that if a negative sense RNA is being sensed by the cell, it will immediately degrade them with the nucleases, which is RNases. And that's one of the reasons that they do have these coatings to protect them. On the other hand, you can see that there are some exceptions, like in coronavirus. They are not naked RNA, but they have a nucleocapsid. One of the reasons may be that coronavirus genome is the largest genome of any RNA viruses. It starts around 28, 29 kilobase. There are usually within 32 kilobase, but there are some which is almost close to 40 kilobase long. There is no RNA virus with such a large genome. After any genome which is bigger than that size, it becomes a DNA virus. And the retrovirus is also another exception, where the RNA is a plus-strand RNA, which is coated by the protein, but it is converted into the DNA by the cellular reverse transcript, by the viral reverse transcriptase. And it's a different mechanism, will be a different topic. We are going to talk about that. On the other hand, the double-stranded RNA genome do contain RNA-dependent RNA polymerase in the particle. It is inside the particle. And here you are seeing the double-shelled or double-capsid reovirus. But it also contains this double-stranded RNA is also naked RNA. So your question will be here, okay, all right. Here you have negative sense RNA. That is, you are saying that RNAs will degrade that in the cell. That's why it is coated. So what about in here? Double-stranded RNA is also not seen in the cell, so that may be degraded by the cellular RNases as well. But the interesting fact that we are going to see that this double-stranded RNA never leaves this core. the mRNAs are going synthesized by the RNA-dependent RNA polymerase, not in the cytoplasm, but inside this core particle in the cytoplasm. And the mRNAs come out of these portals, which we call as the hollow spike proteins or Tourette proteins. They come out through this. And that's the reason that as the cell don't see these double-stranded RNAs, they are also naked RNA and not protected by the proteins. All right, so after that, we are looking at the nucleocapsids, that what we were talking about, where the RNAs are covered with these proteins. And these are the hallmark of the minor strand RNA viruses. And here we are looking at the molecular structure of the VSV nRNA. What we mean by the nRNA, right here in the yellow is the negative sense RNA. And this is the N protein, which stands for the nucleocapsid protein. And you can see that it's a bilobed structure. The N protein is a bilobed structure. And in between these two lobes, there is a groove in which the RNA is actually located, and they are very well protected. And you can see that they are the RNA is packed inside the viral particle. In this is a helical symmetry. You can easily see very, very well protected. And on top of that, you already talked about that on top of that, there is also a matrix protein, which helps in packaging that. And then you have the membrane with the glycoproteins. So very protected nucleocapsid. On the other hand, here you are seeing the influenza virus example, where you can see that this is the PB1, PB2, and PA, and these three subunits of proteins make the RNA polymerase or RNA-dependent RNA polymerase. And here you're seeing the NP protein, NP, again, nucleocapsid protein, just N protein here, nucleocapsid protein. Here, NP stands for the nucleocapsid protein. And it also enwraps the RNA, but here it is a little bit more superficial, doesn't have a groove like this. So in vitro studies, the npRNA is a little bit, this RNA is a little bit more RNAs susceptible compared to the VSV mRNA. And you can see that how is the organization in the viral particle. You can see that this is actually a looped right here, the negative sense RNA. At the end of it, each of the segment will have an RNA polymerase attached to it. So the question is that you may ask that the same way that, okay, this is very protected. If you are saying that this RNA is not much very protected, then what happens when it infects the cell? Now, if you remember that, again, you do not have to remember for this chapter, that the VSV, the nucleocapsid RNA, this is delivered into the cytoplasm, and the mRNA synthesis actually happens in the cytoplasm. Genome replication, mRNA synthesis, both happens in the cytoplasm. Whereas for the influenza virus, it is different. These genome segments are delivered directly into the nucleus. And nucleus is devoid of these RNases because there are lots of RNA synthesis going on in the nucleolus of the nucleus. And these are safeguarded from the RNases which are present in the cytoplasm, but not in the nucleus. And that explains those things. So everything does have a reason for that. And what you are seeing, that these are the charge residues in the red and the blues. And you can easily see that the nucleic acid, the reason that it is nucleic acid, because it is actually charged, negatively charged. And what you are seeing, that's the reason that they are close to this blue residue, which are the positively charged amino acid, because the opposite charge actually attract. All right. So next, what we are going to see, that all these RNA viruses actually do form extensive secondary structure. We talked about that. And here are some of the examples of what kind of structures they actually form. So what you can see, that if they have the complementary bases in proper places, they can form this stem, hairpin, and there are many names of these different structures. One of the interesting structure also formed, which is known as a pseudoknot, and we are going to see this in one of our example, which is the poliovirus replication. You can see that here in the stem of a stem loop, you have the complementary bases, so it makes a loop. But on top of the loop right here, this bases do have complementary bases downstream the stem right here. So what happens that this loop actually falls back on top of that like this and forms a structure like this. And then if this actually folds over, think about that, this portion actually folds on top of that. So you just fold it on top of this, you get a kind of a pseudoknot kind of structure. And this is a typical structure that we see in the poliovirus, and this is an extracrystal study, extracrystallographic molecular model of such a pseudoknot. And they all have implications in regulation of the replication that we are going to see, or the synthesis or processing of the RNAs. So one of the rules for the viral RNA synthesis is almost similar to the DNA replication as well, but it's just a little bit different in some of the cases, that the RNA genome must be copied end to end with no loss of nucleotide sequences. And sometimes what happens, we all know that there are telomeres at the end of the chromosomes, and we sometimes do see that the telomeres actually do shorten. So for, say, human genome, we do have loss at the end of the chromosomes, whereas for the viruses, there is absolutely not a loss of a single nucleotide, and the entire genome from one end to the other end is copied. And the viral mRNAs must be produced that can efficiently translate it by cellular protein synthesis machinery. And this is basically the central theme of Baltimore's scheme that we already see, that the mRNA is at the center of that scheme, and you already know this. So now we have some of the universal rule of an RNA-directed RNA synthesis. Also, we talk, the same thing is said in RNA-dependent RNA synthesis, where RNA is a template. On top of that, an RNA is made. There is no parallel of this in the cell. The cell can make only DNA-dependent RNA, but never RNA-dependent RNAs. There is no cellular machinery that can do that, or enzymes that can do that. So first thing, that just a couple of things here, that RNA synthesize initiates and terminates at a specific site on the template. This is important. And the RNA-dependent RNA polymerase may initiate the synthesis de novo. This is like the cellular DNA-dependent RNA polymerase, or may require a primer. The cellular DNA-dependent RNA polymerase in the cell doesn't require a primer. It can use the de novo. But remember that during the DNA replication, the DNA polymerase actually does require an RNA primer for starting the synthesis. But that's an entirely different ballgame. We are talking about here the RNA polymerases, which doesn't require a primer in the cellular environment, eukaryotic or prokaryotic. But in the virus, it could be either doesn't require a primer or may require a primer. And other viral and self proteins also may be required. This is also another important thing, that there are sometimes host factors involved. That means the RNA-directed RNA polymerase is done by the viral RNA polymerase, but for the functioning of that, some of the host proteins may be involved in there. And the next one is that the RNA synthesized, template-directed stepwise incorporation of NTPs. Remember that these are nucleotide triphosphates, very different from the dNTPs. And very carefully remember that sometimes there could be some trick question in your quizzes where actually it gives you all the answers with a dNTP, which is deoxynucleotide triphosphate, very different from the NTPs, which is a nucleotide triphosphate. When you are saying NTPs, that means your ATP, CTP, GTP, and UTP. These are your 4 NTPs. These are the building blocks of your RNA. When you are saying dNTPs, these are DATP, DTTP, DCTP, and DGTP. So they are all deoxy. So the NTPs has the ribose sugar, whereas the dNTPs have the deoxyribose sugar. And this has been completely elaborated into the supplementary material that you can take a look at. And there are some non-templated synthesis also that we can actually observe. So the next thing is that this is what we talked about, that what we mean by the de novo initiation. So think about that suppose this is an RNA right here. It's a 3' to 5' RNA. Suppose this is a negative sense RNA, and you are trying to synthesize a plus sense RNA on top of that. And right here, this is actually the first N2. This is the first base right here, and N1, N2. So what happens is that NTP directly binds to the first base right here, complementary NTP, and then the second base is attached to this first one, and the chain grows on so far. So there is no primer needed, just directly the first base is added directly to the complementary base on that. So this is your de novo 3-terminal initiation. On the other hand, the primer dependent initiation, sometimes what can happen, that there could be a terminal protein, and this terminal protein can have a phosphodiester linked some of the nucleotides attached to that. Say, for example, we will see that there will be 2 U's attached to the terminal protein in the poliovirus. And on top of this, so you already have a primer. On top of this primer, the NTPs will be added and the chain will grow. And this protein will go off at a certain point of time. So this is also another way that the primer is dependent on a protein. On the other hand, there is also another very interesting thing that we are going to see, that a 5' capped primer can be used to synthesize or start the addition of the NTPs so that the chain goes along. And we are going to see that this is a cap. So what is a cap? Just in a short, and you can just, again, directly, the detail is not needed, but you can just read and follow this link into a wiki page, that cap is a special structure at the 5' end of the RNAs, mainly in the cellular mRNAs and the viral mRNAs as well, many of them. So the cap, the first base is a guanosine or a G, and it has a modification at the 7-methyl base, and this is an inverted, it is incorporates as an inverted form in the first base. And the second base is usually a G, and the third base is usually an A. And in those, there is, in the ribose sugar, the second carbon is methylated, which we call as a 2' O methylation. It may be too much to take right now, and it is not needed. You can just follow this link and just take a look at the cap, just an overview of idea that what it is. So just think here that the cap is a special modification at the 5' end, and why it is important. This is important because there are 2 things. One, if you have a capped RNA, that is resistant to RNase degradation. That's number 1. So mRNAs will not be degraded or touched by the RNases present in the cytoplasm. That's number 1. Number 2, this is essential for the ribosome to understand that this is a message or an mRNA, and it is translation ready, and it will be translated. So these are the reason that you have the caps. So your question would be that here we are not seeing any cap. Sometimes the viral RNA-dependent RNA polymerase also do have these capping function, which they contain the guanylyl transferase or methyl transferase function built into the RNA polymerase. They can put a cap. In some, there may not be a cap, and an uncapped RNA can be also translated, as in the case of a poliovirus, by a special mechanism, and we are going to see that. All right. So the next thing that we are looking at, the sequence relationship among the nucleotide polymerases. And again, you do not have to memorize these things, just a couple of things, take-home message in the sense that there is this GDD, which is the glycine aspartic acid residue. In all plus-strand double-stranded RNA viral polymerases have this one. And right here, you can see that this is a conserved motif right here in the yellow. This is where you find the GDD motif. And there is an aspartic acid in the reverse transcripted and segmented minus strand viral polymerases, whereas it is slightly differentiated into the glycine aspartic acid asparagine in non-segmented minus strand RNA polymerases. So you can easily see that there is a very good sequence conservation of the catalytic site, and we are going to see that what it means. So what you are seeing that this 4 RNA-dependent RNA polymerases have these motifs, which are A, B, C, D, E, and these are the conserved motifs on RNA-dependent RNA polymerases. RNA-dependent DNA polymerases, we are going to see those as well. They actually do have these conserved motifs as well. So DNA-dependent DNA polymerases, which are just the replication enzyme in the cell, they also do have these conserved motifs. And DNA-dependent RNA polymerase, which is in the cell, these are the ones which actually do make the DNA-dependent RNA polymerase make the mRNAs into the cell. This is normal one. So your question is that what is this RNA-dependent DNA polymerase? Remember that this RNA-dependent DNA polymerase is nothing but the reverse transcriptase of the retroviruses. They also do contain the conserved residues as such. So very specific types. So being said that, if we just see just one of the RNA polymerases architecture, then we will understand that how it actually functions. And the most common one is the poliovirus RNA-dependent RNA polymerase, that extensively studied molecule. So the first thing is that how the catalysis occurs, and we have already seen that the bases or the NTPs are added to the 3' hydroxyl. And the base comes as a nucleotide triphosphate, where you are seeing that this is a typical NTP. You have the base. This could be AUCGs, any one of them, the purines and the pyrimidines. And here you have the ribose sugar. And here is one of the important things that I'm just touching, and this is in your supplementary material, how we number the carbons. This is your first carbon, 1, 2, 3, 4, and 5. It's a pentose sugar. And see that at the second position, you have a H right here, so and a hydroxyl. So in your DNA, this hydroxyl is not present. It is H and H. This O is gone, and that is the reason we call it as a deoxyribose sugar, whereas in the RNA, it is an hydroxyl, and this is very important. On the other hand, in the nucleotide triphosphate, there are 3 phosphates attached. This is the building block that is present in the cellular environment. And these phosphates are known as the alpha, beta, and the gamma phosphate. So what you are going to see that this beta and the gamma phosphates are essential for the bonding formation, but they will be hydrolyzed out and they will just get out as the inorganic phosphate or the PPI. And this is the only one phosphate, the alpha phosphate, will go into the RNA chain. So in the RNA chain, it will be a nucleotide monophosphate always, not a triphosphate. So triphosphate comes in, and these two phosphates actually go out in the reaction. So all the polymerases work by a 2 metal mechanism, and usually these metals are magnesium, sometimes calcium, and these are coordinated by those aspartic acid residues that what we have seen. So how this actually works. So what you are looking at here, that this is our template, and this right here is a template, and this one is the 5' to 3' direction. The synthesis is happening. Remember that all DNA RNA polymerases always work from the 5' to 3' direction, where there is an open 3' hydroxyl at this end where a new nucleotide can be added. It cannot be added at the 5' end because there is no open hydroxyl. So right here, where it is added. So you can see that right here is the hydroxyl end, and that will be added by the next base, which is coming in. So why I marked this like this? So if you just carefully look at that, this is the base, ribose sugar, and the phosphate. And right here, this is your base. This is your base. T is the base. This is your ribose sugar. It is just upside down right here. You can easily see. And then right here, upside down means like sideways. This is your alpha phosphate. This is your beta phosphate. This is your gamma phosphate. It is exactly the scenario like this. So what these two things are actually doing. So this is the aspartic acid from the motif A. Remember that although you have a specific sequence of gly, asp, asp, but this GDD motif, this one is not from that GDD. This is an aspartic acid from the A motif. And if we just go this aspartic acid comes from the A motif, another aspartic acid comes from the C motif right here. So what you are seeing, this asp-C means that this is aspartic acid from the C motif, and this one is from the A motif. They are coordinating two magnesium ions. And when these two magnesium ions are coordinating this incoming triphosphate, what you are seeing is this right here, this phosphate is having a nucleophilic attack from this right here. And this will also, so this is basically a deprotonation actually going on. So when this actually goes on, this structure is stabilized by this second magnesium, and this actually helps to keep that transition in the molecule in the transition state so that this reaction can occur. So that's the reason that these two magnesiums are essential to keep this alpha, beta, gamma phosphate in position so that this reaction can go on. And that's basically, and again, we will not go into the organic chemistry. If you just know the overall mechanism by these two metals, that's good enough. So this beta and the gamma phosphate will actually go out as the inorganic phosphate, and this will be the only alpha phosphate will be in the chain, just like you are seeing right here. So that's your polymerase catalysis. So here is an interesting thing, that this is a structure of the active site of the poliovirus RNA-dependent RNA polymerase. And this gives you an idea that why the poliovirus or any RNA-dependent RNA polymerase will have the specificity for the NTP or nucleotide triphosphate, but not DNTP. It will not put a building block of the DNA, which is a deoxyribose, but will always put a nucleotide triphosphate with a ribose sugar in it. So what you are seeing, that this is a residue right here, D238, and what the glycine aspartic acid is doing right here, it is forming a hydrogen bond with this hydroxyl. So if you just want to put this in the perspective, it may be a little bit obscure, but you can see that. I will just explain right here. So right here, you have a 2' hydroxyl right here. In the DNTP, this O is not in there, but let's take a look at the structure first. So this is a nucleotide right here. This is the base, and in the structure, that's the base right here. Then, so it is the opposite way. And just think about that if you just make the molecule other way. And right here is the ribose sugar. Right here is the ribose sugar. And then you have the 3 phosphates that are coming in, and these are the 1, 2, 3 phosphates. So that is being incorporated into that structure. So what you are seeing, that this structure right here is the 2-hydroxyl end right here, and that can form a hydrogen bond with the aspartic acid at the 238 position. What happens that instead of this, if you have a DNTP with just an H, without that oxygen comes in, this hydrogen bonding will not be able to form. And this is this subtle difference of just one oxygen right here completely changes the specificity of this. So you can easily change, so that's the reason it's very specific. So what happens in a DNA-dependent RNA polymerase, what happens, or DNA polymerases right here, which can incorporate a DNTP, here usually you have a tyrosine residue. So recombinant RNA polymerases or engineered RNA polymerases of these viruses have been made where they replace or mutated this residue with a tyrosine, and now it can incorporate DNTP into the growing chain. And this is very weird experiment that has been demonstrated, that why this very specific specificity exists just due to this ribose sugar and this hydroxyl group. All right. So this is the part of the structural biology that you can actually see this interactions directly to explain that why something is actually happening. So right here, this is, I'm sorry, this is, hold on. This label is a little bit off. Just give me 1 second. I just make the correction right here. So that's the structure at the active site, and That's about it. So now what we are looking at, that this is the structure of the poliovirus RNA-dependent RNA polymerase. And this RNA-dependent RNA polymerase is named as a 3D pol. And it's a famous molecule. A huge amount of work has been done on this. And we are going to explain that why it is named as a 3D pol. So that's a mystery right now, but we will explain. So structural biologists actually try to think, and they think that all the polymerases or polynucleotide polymerases, that means that DNA polymerases and RNA polymerases, they all have a right-handed structure. So in the right-handed structure, you have a thumb, then you have a palm, and then you have a fingers. And that actually is kind of a structure that all the polymerases actually have. And the catalytic site is present in this palm region. So what you are seeing right here is almost similar. Right here, you have the thumb, then you have this beta strands, which make the floor or the palm, which is the catalytic site. Right here, you have the catalytic residues. And then right here, that's the catalytic residues that you can see. And right here in the back, that is the finger. So you can easily see the motifs that you have, specific motifs. All the colors actually do not correspond to that. This cyan is corresponded to that grayish structure. So all of them are not the same. But you can see this purple is right here, and the red is right here. The yellow is right here, and the green is right here. So, but the cyan is replaced by this, and it was just not in the correct sense. But this is the overall structure of that. So now what you see, that if you think about this, that if you actually close this hand, then on the top right here, you will have an opening. And when you are just clenching your fist, in between your thumb and the fingers, there will be some opening. And this will make the back side of the hand. And you can think that in between the fingers, you can have some small openings in there. And those things are important in the functioning of the polymerase. And we are going to see that in a second. So let's take a look at that, how these 3D pol or the RNA-dependent RNA polymerases of the poliovirus actually do work. So the first thing that what we are seeing, that here we are looking at the RNA polymerase, and again, remember that this color code right here is very different from this one. They are not the same, so do not compare those. So right here, what we are looking at is the thumb right here. These are the finger. This is actually making the palm, and you are looking at this structure right here. So right here, this is the red position is your thumb. This is the back of your finger right here, which is making this green part, and then your palm is basically this region. So the top is open, which is this opening. If you have the hand like this, so the top is open, and what you are seeing, that the active site in the purple, which is like deep into this on top of this yellow region, which is basically denotes the palm, and that's the active site deep inside. And this right here is the cyan RNA that is being shown here. That is the template, and the template goes through this top pore or the top hole into the active site. So once it goes into the active site, it hits the active site and it makes a 90-degree turn, and then it exits from the front. And in that process, the product is actually made in the active site, and the product also leaves from the front. So you can easily see that this is the top through which the template came in. Right here, just imagine a little bit of imagination, that think about that there is some pore right here. And through this region, the template that is being used and the product will come out. They do not come out as double-stranded RNAs. They keep them separate. And this is the top through which the RNA template is going in. So your question could be that why we are seeing right here the green RNA or the product from the top as well as a template. Now, I have explained that in the narrative below in the notes. Remember that these are all crystal structures of these, And when they were crystallized, you make a template RNA, you make a product RNA, you purify these proteins, and then you co-crystallize all of them so that you think that they will all bind in proper places along with the NTPs. Now, remember that the NTPs are actually replaced by some of the analogs so that the reaction doesn't go in. The NTPs will bind and it will stabilize the RNAs, but it will not be able to synthesize farther because the nucleotide analogs are similar, but it will not able to form the bond or the phosphodiester bond for the synthesis. And as those things are being done, this is artificially made and it is just shown in the structure. But that is not that will not be present in a native condition. So it will be just like this. So that comes from the front. Now the interesting part is that, think about that, this is the back of your hand, so the palm and back of your hand, and think about that in between your fingers you have a little bit of space. You can just imagine like this. And those spaces, think about that, those spaces are these tiny spaces. These spaces are much larger. This is a much larger space for the template to go in, much larger space for the template and the product to come out. But at the back, you have a very tiny opening, and the nucleotide triphosphates actually diffuse into the RNA polymerase active site through these holes. So this is how it actually works. So this is the molecular detail that the structural biology can actually provide. And pretty much all RNA-dependent RNA polymerases actually work in almost the similar fashion as well. So now what we are going to do, so that's all about the general scheme or the general theme about this chapter, the general principles. Now we will look at some of the synthesis strategies and example. So before we start, any specific questions? Anyone? So you can stop me and just say anything that you wish to.
Just a random question, just to keep it straight in my head. So
Yeah.
DNA polymerase do not have 238 aspartic acid, and that's just like the main difference between RNA and DNA polymerase is that DNA polymerase doesn't have the 238 aspartic acid.
Oh, no. Hold on 1 second. Yeah. So just give me 1 second. I just replace, just give me 1 second. Good question. I will just answer that in a second. All right. Yeah, that is absolutely correct. So you can see right here, the main difference is that in the DNA polymerase, this D238, which is aspartic acid, is replaced usually by tyrosine, so that this bonding actually cannot occur. That makes sense.
And the like coordination complex that forms with magnesium, that's not specific to
most of them. Yeah. So the coordination complex actually forms with the magnesium. And along with that, the interesting part of that as well, that sometimes there are some specific polymerases where the calcium, which is a divalent cation, that can also coordinate. So that's why we just said that instead of just saying magnesium in our heading right here, we actually just mentioned right here is a two-metal mechanism, and usually it's a divalent cation, and magnesium is very common. There are some examples with calcium. There are some examples with manganese as well, Mn2+. All right.
Gotcha. Thanks.
Yeah, sure, absolutely. Any other questions, folks? If you have. You guys are good with the slide, right? That I just turn it on and off a couple of times. Okay. All right. So let's go to the next part. And now things will get a little bit difficult in the sense. So just bear with me with that, because we will go into a little bit of hardcore molecular biology to some extent. So the first one is the Baltimore class 3. So we'll start again. We will start right here, the class 3, then we'll discuss the class 4, then we'll go to class 5. So the first one is the Baltimore class 3 dsRNA virus, and the example is the Rio virus, and it is belongs to the Rioviridae family, and double-stranded RNA genome, usually Rioviridae is 19 to 32 kilobase long, and they have 10 double-stranded RNA segments. And if you can see that they are divided into large, medium, and small, and the 5' to 3' is a capped RNA right here. You can see that it is a capped RNA, so this will be very similar to an mRNA, and you have the complementary negative strand RNA as well. So from this double-stranded RNA, the mRNA is formed. So the mRNA will be templated on the negative strand, and this will be formed. This will come out in the cytoplasm that we are going to see and form the proteins required for doing all the function, and the viral particles will be made. And the interesting thing, what happens, we are going to see that as the viral particles are being made, or the viral core is made into the cell, the mRNA, again, remember that the mRNA is going to get packaged into the virion or the core. And along with that, it will also package the RNA-dependent RNA polymerase, which will make the double-stranded RNA from the packaged mRNA. And that is the way this double-stranded RNA is never exposed into the cellular environment. It will just be clear in a second. So here is the strategy. So here you can see that this is the double-stranded RNA. So in one scheme, that what is happening, that just think about that this is your minus strand right here, 3' to 5'. It is just in the opposite way. So just usually the convention is we just always read from left to right, but in the book they have used this. So this is a 3' to 5' end, and on the 3' to 5' end template, the mRNA, which is exactly like this strand, will be made. 5' to 3', 5' is capped. This comes out of the virion and will be translated into all the proteins. These are the structural proteins, non-structural proteins. The non-structural protein means that there are some proteins which help in the assembly regulation of the virus, but they are not a part of the viral particle. We'll see that in a second. So, So that's your mRNA synthesis. The replication is the same. The first step is the mRNA. So you can just easily replace this with this one. It's basically the same thing. But what happens that a portion of the mRNA, which is synthesized just like this, is saved. And once these proteins are formed, it forms the viral particle and package some of the mRNAs right here. And this double-stranded mRNA actually remains inside the virus, which makes the mRNAs. And these mRNAs will be packaged into the new viruses. And inside the viral particle, the double-stranded RNA will be formed back again, because this will be now the template to form the negative strand. So let's take a look at the life cycle, and that will make the idea. So we are going to see these the reproductive cycle for each of the viruses. And remember that we are not that much interested in the other steps like the entry and assembly. We're going to discuss those, but we are mainly going to talk about the RNA-dependent RNA synthesis, which is in this context. But as we have already talked about this in the structure of the Rio virus, this is just giving you an example that how exactly this mechanism actually happened. So Rio virus enters through a receptor-mediated endocytosis through a cluster-encoded pit. You can see now you have the endosome. Now the endosome pumps the H+ ion, become acidified, fuses with the lysosome, and these lysosome will digest the outer coat or the outer capsid. Right here is the protein, and will expose some of these hydrophobic residue proteins, which is called the ISVP, or the infectious subviral particle. This blue proteins right here do have the hydrophobic residues, which were covered by these orange proteins right here, and now they are exposed. They will fuse with the membrane, and the infectious subviral particle will penetrate through the endosomal membrane into the cytoplasm like this. In this process, these hydrophobic residues or the blue proteins will be stripped off, and Another very interesting thing is that what you are seeing, that we are going to see that in a next slide also in detail. You can see that right here, there are some spike proteins. We also call them as a Tourette proteins. These are hollow spikes. There is a hole in the middle, and that spike or the hollow is capped by another protein. There is a cap, so it is closed. When the reovirus, this infectious subviral particle, penetrates the endosomal membrane and goes into the cytoplasm, this cap is actually removed. So you can see right here, this blue protein, which is responsible for the fusion or the penetration of the endosomal membrane, is removed, and this cap from these turrets or the hollow spikes are removed as well. This is the viral core or the inner capsid. And now what happens inside, you have the double-stranded RNA. As soon as these caps are removed, it changes the conformation, and the RNA-dependent RNA polymerase become active inside the virus, and it starts making the mRNA inside the virus, which leaves the viral particle through these portals. These are the opened up Tourette, hollow Tourette proteins, or the spikes, which were completely covered right here. So now these mRNAs are coming out, and these mRNAs will make the proteins required for the virus, and the virus will pack these mRNAs, and the virus will. So this is the process. The virus will make this particle right here. So what you are seeing, that these are the mRNAs, and these mRNAs are now covered with these this is the viral capsid. This is covered right now. So this is the intermediate form of the virus, with this dot means that they are all mRNAs right here. So now what happens, that inside the virus, as these mRNAs are in there, the virus will assemble to form all other proteins will be formed. And inside the virus, the packaged RNA-dependent RNA polymerase will convert these mRNAs into the double-stranded RNA. So this double-stranded RNA never comes out of the particle. So that's what it is that we talked about right here, that the mRNA will be packaged into the intermediate particles that will be converted into double-stranded particle as the virus matures. So this is the interesting part, that how it can happen. And also these particles right here, where you have these mRNAs, some of these can be converted into the double-stranded RNAs, which will form more cores inside the cell, and these will also pump out more and more mRNAs. So that's the reason that it has been shown this way. So it's a very interesting strategy. I just wrote it down detailed in the narrative. It may take a moment to understand the process, but just read it step by step, you will understand the process. Again, just to recap, because I don't want to rush through this, that this inner core brings the double-stranded RNA, which is making the mRNA that is coming out in the cytoplasm. That's making the viral proteins. And these mRNAs will be packaged with the viral protein to make the core. This is the core, which will form, again, this structure right here into the cell. Many of these will be formed, which will make the double-stranded RNA inside them, will make more and more mRNAs to make this cycle go on. So this will go on a cyclic fashion. And out of that, some of them will mature into the virus with the second coat and come out of the cell. All right, so here is the detail of the genome mRNA synthesis and the translation. You do not have to memorize any of these name of the protein. Just remember that the largest protein, which is the lambda-3, and another protein mu2, they actually made up the RNA-dependent RNA polymerase. So this is the real RNA polymerase, which actually synthesized the chain. And this mu2 protein is the guanylyl transferase, or the capping protein, and this makes the mRNA caps. And per reovirus particle, there is only 12 copies of the RNA-dependent RNA particle, and they are placed under the 12 vertices of the icosahedral symmetry. And also what you can see that there are some of the proteins that you can see that mu-NS, mu-NSC, sigma ms. NS means that these are the non-structural proteins, means that they are produced in the cell, but they are responsible for regulation, assembly, and packaging of the virus particle, maturation of the virus particle. But these proteins are not present inside the virus particle. That's why we call them non-structural protein. The others are all structural proteins. So here is an overview of that will actually give you the idea. This is the schematic, and you can see that this is the spike, hollow spike protein, or the Tourette, which is capped by this sigma 1 protein. So this is a Tourette. So what we were talking about, and that is how it's named. So that is the spike or the Tourette is the same thing. So you can see that right here, the outer capsid is made up of this sigma and the mu1, and the inner capsid is made up of this sigma 2 and the lambda 1. And under each of these 12-fold vortices, although it is in 2-dimension, the RNA polymerase actually stay right below right here, these hollow Tourettes, which is made up of the lambda 3 and the mu2, the RDRP, and the guanyl transferase, which makes the RNA polymerase. So what you are seeing that if we just think the strategy that what happens, the reovirus once goes into the endosome, acidic endosome, the sigma 3 proteins right here, these are digested by the lysosomes. And it exposes these blue proteins, which is the mu1, and these have the hydrophobic residues on top of that, through which it is able to penetrate the endosomal membrane. And this is called the infectious subviral particle. As it penetrates the membrane in that process, this mu1 is stripped off, and right here, the cap, right here, this cap, which is also an attachment protein or for the receptor-mediated endocytosis, this sigma 1, that also comes off. So right here is the core where the Tourette protein is now empty, and you have the double-stranded RNAs. Once this sigma 1 goes off and the hollow opens up, the RDRP is activated inside the viral particle, will make the mRNAs, which will come out into the cytoplasm through these portals. So that's basically the overview of that. It's a very elegant mechanism and amazing little molecular machines, what they can do. So here is a little bit of fun what I have done. I have to share the desktop actually. All right, so this is a link that you can play with. So this actually is the protein data bank, which is a PDB, and you have all the different structures of in here. So what you are seeing is the molecular structure of the reovirus core. And I'm just putting this one in a perspective sideways, say for an example, right here. And right here, this protein right here in the pink, this is the Tourette protein, and you have one of them in each of the 12 vertices. So you can see that on the side view of that, if you just like to see that right here, this is the Tourette from the side view. And if you just look at from the top, you can see that right here in the middle, right here, you have the pore or the portal through which the mRNA comes out. And this is actually capped by the sigma 1 protein, and that is completely capped and in the intact particle. But when the core is delivered, this opens up, and through which the mRNAs come out. And the RNA-dependent RNA polymerase is present at the base, deep inside the core, below this base. And also, there is a very interesting thing that actually happens. As you have 12 of these, one in each vertices, each of the vertices is attached with one segment of the double-stranded RNA. So each one of them, say 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So each one of them is under that. So this is a very interesting thing that actually happens. So let's go back to, all right. So what happens, so you can easily see that you have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So you have 2 extra, but under each vertices, there is one one of these double-stranded RNAs attached. So what happens, interestingly, for that reason, there is no reovirus where you can find more than 12 segments of RNA. You can maximally, there could be less numbers. So your question could be, if there is 10, what happens to the 2 extra portals or something like that? We really do not know. Maybe empty or maybe randomly added with any other RNAs out of those. We really do not know. But the number of RNA segments cannot be more than 12 because there are 12 turrets at each vertices, and each are associated with one of the RNAs. And below right here, you have the RNA polymerase, which actually makes the mRNA, and this mRNA goes out to the specific portal. So you can also think that each portals are the passage for each specific mRNA out of the double-stranded RNAs as well. So we will actually, it's 9:12, we will actually stop right here. And I just don't want to rush through that material. And we will start with the poliovirus in the next class. And that's pretty much it. So any questions? So you guys are all in this screen now. Any questions that you may have? No? Are you guys good? Any specific question that you may have? Are you guys good? Okay, all right. So maybe a little bit of a steep curve. It may look like this second module is a little bit intimidating. Don't worry about it. You will have enough practices. We'll have enough review, and you guys should be fine. And again, yep, that's pretty much it. So we are not giving you any for the graduate students and the honor students. I am not giving you a topic yet, and this weekend you will not also have a discussion done. You need to digest this material before you dive into some of the discussions. So your first discussion for the second module will be on the next week. But this weekend you will have one quiz due on this one. All right, good, everyone. So we are 2 minutes early. That never happens. All right, but again, let me know that if you have any questions regarding test 1, if you want to review, if you need office hours. I know I already got some emails. Just take a look. Thanks a lot, folks. Yeah, appreciate it. All right, bye-bye. We'll see you Thursday.